Hello, I am Victor Grentzinger, and I'm pleased to present to you the advances of my PhD project about lifting the veil on challenging medically relevant genes or CMRGs. These genes of clinical interest have a segmental duplication of part or the totality of a sequence, such as one or multiple pseudogenes, or a variable non-tandem repeat, or VNTR. This is of major concern because this leads to incorrect characterization of the region of interest when sequencing with Sanger or short-read methods. For short-read sequencing, this is mostly due to limitation from bioinformatics tools, as the reads are ambiguously aligned to multiple parts of the genome. To determine if we could bypass this limitation, we investigated the use of long-read sequencing in a clinical setting with a focus on the PKD1 gene, responsible for autosomal dominant polycystic kidney disease or ADPKD, which has multiple pseudogenes, and the filaggrin gene, which has an impact on atopic dermatitis or AD. For filaggrin, it has a VNTR on its third exon and 3 different known alleles. So you have on the right a simplified representation of our Nextflow pipeline, where each tool is containerized by using Aptainer for reproducibility. So, for PKD1, we created a cohort of 34 ADPKD patients with a total of 56 pathogenic variants confirmed by Sanger sequencing. We then did 2 different runs, one with whole exome Illumina sequencing and the other with amplicon nanopore sequencing. All variants confirmed by Sanger sequencing have been retrieved for both runs. However, we found that for our amplicon nanopore run, we have a better allelic frequency for heterozygous variants compared to whole exome Illumina sequencing, as represented here. Therefore, out of the 2 methods, our long read one is the best to use for characterizing the PKD1 gene. For filaggrin, or FLG, I have downloaded 1,134 publicly available long-read whole exome sequencing data. And for each sample, I have labeled the population of origin and only kept samples with a coverage of at least 2x on the third exon, which gives me 1,011 samples of 28 different origins. After analyzing the data, I have found that not only this gene has 3 different alleles, but there is also 6 novel alleles that have never been described in the literature with a large change in the number of repetitions of these genes. When we look at the frequency of these novel alleles, while there is some in all populations, they are mostly found in African population, as you can see here in yellow, where more than half of the samples that has been analyzed have at least 1 novel allele. This is mostly due to the fact that these alleles were invisible before when we were using short-read sequencing, but they were also hidden by the fact that there are few genetic studies of atopic dermatitis in African and African descent population compared to European and Asian populations. So, this work not only shows that for some genes, the use of long-read sequencing in the clinical setting is possible, and even better than short-read sequencing, but also that we are able to find new genetic diversity completely obscured by the sequencing method when we look at these genes with the right tool. This can be of importance, as a study showed that the length of the filaggrin allele can have an impact on the severity of atopic dermatitis. I also looked at the MUC1 gene, responsible for autosomal dominant tubulointerstitial kidney disease or ADTKD, which also has a VNTR in its sequence. the same as filaggrin. However, the limitation for this gene is different from filaggrin, as it is now the reference genome which is a limitation in correctly characterizing the complicated sequence of MUC1. To overcome this, we believe that we should create a pan-genome of the MUC1 genes and align the long-read data back to these pan-genomes, as it could improve results. I want to thank you for your attention, and please visit my LinkedIn page by scanning the QR code on your right. Thank you.